Hello, today's topic is cellular aging under the chapter Cell Injury. So, the most widely accepted theory of cellular aging involves free radical mediated damage. So, why does cellular aging occur? It occurs as a consequence of progressive accumulation of cellular and molecular damage due to genetic and exogenous influences over a prolonged period of time. First, we start with telomeres. What are telomeres? Telomeres are actually short stretches of DNA situated at the end of chromosomes. So, these function mainly to protect the chromosomal ends from fusion and degradation, and thus it ensures complete replication of the chromosomes. So, as the cell continues to divide in mitosis, progressive shortening of telomere occurs. Eventually, it leads to cell cycle arrest. This flowchart shows how it actually occurs. Here we can see shortening of telomere occurring with progressive cell division, and it leads to activation of tumor suppressor genes. Tumor suppressor genes, that is p53 and retinoblastoma gene, which leads to cell cycle arrest. Cell cycle arrest, which in turn leads to replicative senescence. Replicative senescence refers to a terminal non-dividing state. A terminal non-dividing state which eventually progresses to cell death. So first shortening of telomeres occurs, then activation of tumor suppressor genes occurs, then cell cycle arrest occurs, leads to replicative senescence, that is the cell enters into a terminal non-dividing state and eventually triggers cell death. Next, about telomerase enzyme. This is the enzyme responsible for maintaining the telomere length. So these are actually reverse transcriptases. That is RNA dependent DNA polymerase, which uses its own RNA as a template for adding further nucleotides. So this telomerase activity is absent in normal somatic cells, but it is expressed in germ cells, and some low levels can be expressed in stem cells also. So this diagram shows telomere length has been plotted against the number of cell divisions. So here, as we said, in germ cells, there is occurrence of the telomerase enzyme. The telomerase activity of the enzyme is present, as a result of which the telomere length is maintained. And in stem cells also, to some extent, the telomerase activity is present, and that's why they also maintain their length to some extent. But in somatic cells, telomerase activity is almost nil, because of which the telomere length decreases with the number of cell divisions. And so, at a certain point, after a certain point, it decreases to such a level that growth arrest occurs. Whereas here, note that in cancer cells, at some point, there occurs reactivation of the telomerase enzyme. This reactivation spells trouble, because then the cell can continue to divide without any restriction. So the changes seen during aging include, one is increased free radicals and telomere shortening. Apart from that, there is decreased expression of the insulin growth factor 1 gene, that is IGF-1 gene. And also there is increased cross-linking of collagen and decreased somatic mutations due to increased somatic mutations due to DNA damage. So these are the changes seen in normal cellular aging. A few words about sirtuins. Sirtuins are substances which play a role in increasing longevity. So what happens is that these are actually belonging to a family called NAD-dependent protein deacetylases. And these play a role in increasing longevity. And certain environmental stresses, such as if we reduce our calorie intake, calorie restriction, that actually promotes the formation of sirtuins. And this is how the mechanism of action of sirtuins, as to how does it actually bring about this reversing the cellular aging trend. So this is one is by inhibiting the metabolic activity, and it reduces apoptosis, and it stimulates protein folding, inhibits free radical damage, increases insulin sensitivity and glucose metabolism. So these are the points by which sirtuins increase the longevity of the cell. So one question, increased incidence of cancer in old age is due to, we already saw that in cancer cells what happens is that unfortunately there occurs reactivation of telomerase enzyme, due to which there occurs unrestricted division of the cell, uncontrolled division. So this is a flow chart to sum up the mechanisms that cause and counteract cellular aging. Most important is DNA damage leading to mutations over a long period of time. Then is cellular senescence leading to cell loss, which also contributes to cellular aging. Apart from this, defective protein synthesis leading to decreased proteins, damaged proteins, decreased cell functions, and all of these processes, that is DNA damage, cellular replication decrease and decrease in proteins, lead to cellular aging. Whereas some environmental stresses such as calorie restriction, nutrient sensing, they activate various growth factors and transcription factors in the cell. So this altered transcription leads to increased DNA repair, increased protein homeostasis, and counteracts aging. So this is a summary of the mechanisms that cause and counteract cellular aging. So here also we see the role of sirtuins. So the main moral of the story is that the most effective way to prolong lifespan is calorie restriction. Thank you.